All right, folks, for chapter 4, typically transcription translation is kind of a hard concept sometimes for students to get. So when we're talking about transcription translation, what we're really talking about is protein synthesis. So how is it that you go from a DNA molecule that has the information to make a protein and actually make that protein? And the other thing you have to think about, the problem that cells have to get around is a eukaryotic cell, right, has the cell membrane and then the nucleus. The ribosomes are out here, and the ribosomes are what make proteins. Well, the information to make the protein is inside the nucleus. So how do we get this information outside of the nucleus, right, and actually make the proteins out here? And that's really what transcription and translation is, right? So how do we get this information out of the nucleus to make the protein out in the cytoplasm? And the two ways that is accomplished is through transcription and translation. Right? So in transcription, the way I typically tell students to think about transcription, when you're transcribing something, you're typically, so if we're not thinking about cells, we're just thinking about, transcribing. You're typically keeping things in the same language, right? So if you have a court that is transcribing something, they're not changing the language. They're keeping it the same so that it can be read later. So inside the cell, what you're really doing is taking, so this is the DNA molecule over here, right? And this is the other side of it, right? How do I know that's DNA? Because it's got the A's, the C's, the T's, and the G's, right? And then this one is the RNA molecule that is being built. How do I know that's the RNA molecule? Well, there's the U's, right? And so anywhere you see a U, that's going to tell you that is an RNA molecule. And so what RNA polymerase does is it comes along, so the DNA gets unzipped. There's mechanisms which take place that cause that to occur. Again, we won't worry too much about that. You can spend a lot of time going into detail on this. So I took a semester-long molecular class, and we spent weeks just talking about all the stuff that happens here. So we're condensing it, right? So we're going to take this double helix, and we're going to unzip it. And you're going to unzip it at a gene that you want to transcribe and translate, right? So a gene that you want to make a protein out of. So to get that to happen, the RNA polymerase is going to come along the DNA, and it's going to make a complementary sequence to that DNA molecule. What do I mean by that? Well, if you remember, A's go with T's and C's go with G's in DNA. Well, with RNA, what you're going to do is you're just going to replace that T with a U, right? So now this is the T in the DNA. It goes with an A in RNA. There's an A in DNA. It goes with a U, right, in RNA. And so in this manner, you're going to create this RNA sequence that we call messenger RNA, sometimes abbreviated as mRNA. Why is it called the messenger? Because it's going to take this message and it's going to leave the nucleus and it's going to travel outside, right? So that is transcription, right? So transcription is just taking DNA and turning it into messenger RNA, and then that messenger RNA leaves the nucleus. Now there's a lot of things that can happen in between there. So for instance, one of the things that happens is called alternative splicing. So for instance, what you can have, so when you take this DNA and turn it into RNA, sometimes what you can have are different kind of segments, like this. This would be the RNA molecule, where these pieces would be called introns, and the reds would be called exons. Introns are intervening sequences, and so what would happen is these introns would get spliced out and removed such that you would have just one piece that would be red here in this situation. Why do that? Well, one of the reasons you can do that is because you could take the same piece of DNA and splice out different parts of it and end up with different proteins. So it lets you have the same molecule, the same part of the same molecule, and get different proteins from that, right? You don't need to worry too much about that, but it's just an interesting bit about how our DNA works. Right, so transcription, take DNA, turn it into mRNA, and then that mRNA passes outside the nucleus and ends up out here in the cytoplasm. Right? Then what happens is this messenger RNA gets fed into the ribosome. The ribosome is kind of a two-part organelle, so it has a large subunit and a small subunit, and then the messenger RNA kind of gets fed through, right? Almost like I used to say the tape through an adding machine, but that's probably starting to be a dated reference there, right? Maybe a rope through pulleys or whatever. I don't know, but it just gets fed. So you can either this talks about going in this direction. Right, so that's saying that the ribosome is going in that direction. The other way to think about it is if the ribosome is going in that direction, the messenger RNA is going in the opposite direction. Right? So the thing that happens then on the messenger RNA in the ribosome is there would be a sequence. Right? So notice this AUG here. Right? That would be the first sequence of the messenger RNA, AUG. And so what happens in the ribosome is this transfer RNA, sometimes called tRNA, is going to transfer an amino acid over. If you remember back to macromolecules, all proteins are amino acids that are stuck together. Right? So the transfer RNA is going to come along, it's going to jump into the ribosome, and it's going to find those first three nucleotides. And those first three nucleotides are going to match up to the nucleotides that it holds. Right? So we call these three on the messenger RNA the codon, and these three on the transfer RNA the anticodon. Right? So if this is the codon, right, what the transfer RNA is going to be would be, remember, like, so A's go to U's, U's go to A's, G's go to C's. Right? So that transfer RNA then is going to have UAC at the bottom. It's going to match up with this UAG, and then it's got a amino acid up top. Right? So again, the transfer RNA comes in, it finds its sequence. When it finds its sequence, it leaves the amino acid behind, and then it leaves. Right? And then the next one pops in, finds its sequence, leaves the amino acid, and then leaves. Right? Such that what you end up making is a chain of amino acids like this that we call a polypeptide. Why polypeptide? Because each one of these bonds is a peptide bond. And so if you have multiples of them, you end up with a polypeptide. Right? And so then, once this polypeptide chain is formed, it's going to start folding on itself and create the final protein that needs to that's being created, right, and going to be used for whatever, right? And so that's kind of transcription translation in a nutshell. So you take DNA, you turn it into messenger RNA, that's transcription, and then translation. So if I talk about transcription as being the same language, translation, you're turning it into a different language, right? So in translation, you're going it from the language of nucleotides to the language of amino acids, right? So from DNA to messenger RNA, and then from messenger RNA to a chain of amino acids or proteins. And that's really, sometimes we call that the central dogma to molecular biology, right? So from DNA to RNA, some say to protein, but oftentimes I'll just say to amino acids, right? So DNA to RNA to proteins, or DNA to RNA to amino acids, right? And that's transcription translation. So I've posted a couple videos for you about that, and then so you can watch those, and hopefully this one might help as well. But as always, if you have any questions for me, then by all means let me know. Take care.